Hello dear students, welcome to a unique type of class called as Advanced Learning. Here, this is to satisfy the needs of certain students who want to know more and more, sometimes in depth, sometimes those topics which are generally untouched in regular classes. So today we'll learn about integrated microbiology with molecular genetics from biochemistry, where we discuss gene regulation. So how do we go about it? First, let us try to understand what exactly is present in case of gene regulation per se. Okay, in gene regulation per se. So before we start, let me tell you, I'm Dr Meenakshi Sundaram, NEET PG educator, and I'm an MBBS MD, and I have gold medals in my MD exams and MBBS exams, and I'm a high octane educator for biochemistry and microbiology over 11 years, and I have successfully placed many students in the prestigious institutions every year, and I've been in Unacademy for the last 2 years. So I hope I'll be able to help you a lot in your preparations. Before I start the subject, let me give you a small introduction to the Plus subscription here. You have daily live classes, structured courses, live tests and quizzes, and unlimited access to all the kinds of Plus subscription classes on all the subjects under NEET PG category. You can use Dr ASM live code or DRASM live as the subject code for getting 10% discount and unlimited access to all my classes exclusively. There is something called as iconic subscription. You get the best of Unacademy and the best of Prep Ladders, video lectures and question bank. You can have live classes and batch courses, live tests and quizzes, and have rapid directory courses. Here also you can use the same code DRASM live to get 10% off on Unacademy Plus subscriptions. You also have a feature called as raise a hand. In your app, you'll be having, when you come into a live class, you will have a small space where there'll be a raise a hand option. If you click on it, I will get a notification and I can send you an invite for talking to me directly. You can accept the invite and you can start talking to me directly. While we are in the live class, I will be able to answer your queries. So what are the special class features? You have interactive live classes. You can go for polls for some special questions. You can have a raise a hand. You never have to miss a class because you'll be getting notifications every time when there is a live class which you're enrolled into, and you can have lecture notes and you can attend the classes anywhere, anytime. And this is how you can go for the Plus subscription. And you can boost your medical PG preparation between 25th to 28th October by subscribing to Unacademy. By buying for 24 months, you'll get 4 months free. And for buying for 12 months, you'll get 2 months free. And those people who get anything subscription about 12 months or 24 months, you'll be also getting a printed notes in the future. So there is an exam day, monthly free grand test for FMG students 2021. The next test is on 31st October, 10 a.m. to 1 p.m., 300 questions. So these are the toppers of the NEET PG from Unacademy. So these are the two different batches happening: Target NEET PG 2022 batch, where I'll be teaching, and a clinical case discussion batch for you exclusively. So let's begin understanding the concepts which we were about to talk. First, in microbiology, we learn something called as bacterial genetics. When it comes to bacterial genetics, we always learn about bacteriophage mediated transfer of information. Let me make it quick with a small introduction here. Say, if a bacteria A is a toxic bacteria, but a bacteria B is a non-toxic bacteria. If the toxic bacteria A is toxic because of a property called as X gene, and if that X gene is not present in bacteria B, then that gene can be transferred to bacteria B with a bacteriophage intermediate. So bacteriophage intermediate can spread the infection, and if it is infecting organism A, and takes the gene from organism A and deposits in B, and what happens to B later, if B from being a non-toxic organism becomes a toxic organism, you call it as transduction. Transduction. Now this is what we learn in terms of bacterial genetics when we want transfer of genetic information. Here I would like to tell you, when a bacteriophage, generally a DNA virus, is about to attack a bacteria, you can expect two kinds of results. Look at this. It can cause a lytic cycle or it can cause a lysogenic cycle. Lysogenic means it is trying to change something. Lytic means it is trying to replicate. I repeat, if a virus is trying to enter into a bacteria, it can either try to change the bacteria into something else, or it tries to use the bacteria to multiply to many numbers, the same bacteriophage. Up to 100 viral particles may be formed at this particular example. Now imagine we are looking at E. coli being infected by a bacteriophage, and that will be referred to as a lambda phage. Now what are the things I would want you to remember? See, you can have alternative life cycles, I told you, right? What are the alternative life cycles? When a virus called as bacteriophage introduced into a particular bacteria, it can take up a pathway where it can actually be going for a viral infection. On one side, it can go for lysogeny. On the other side, it can go for lysis. Lysis is the site where you have replication, and lysogeny is the place where you're trying to integrate with the host chromosome. Now imagine this. If this is the bacteria, if the virus is bringing a new information, and the bacteria will be already having its own information here, and if the bacterial information and the viral information are coming together, then that will be called as lysogeny. So this is the place where you have to understand you will be having something called as a transduction happening. Let me put it in simple words again. Alternate life cycles of bacteriophage lambda can be spoken if infection of the bacterium E. coli by phage lambda begins when a virus particle attaches itself. As it happens, the injection of DNA has happened. Now remember, when it replicates passively as a part of bacterial DNA during E. coli cell division, the dormant genomically integrated virus is called a prophage. At this point of time, you call it as a prophage. Sometimes when the bacterial information has been confused with the viral information, now you will be having many portions of the viral information while the bacteria will be holding on to its host information. Now this can instantly break into pieces. The bacterial chromosome also breaks into pieces. You will be having synthesis of bacterial proteins and synthesis of viral proteins. You will have synthesis of bacterial nucleic acid and viral nucleic acid also. At the end of it, if the viral proteins are attaching themselves and the nucleic acids are packed inside themselves, then when new viruses come out of it, this will be referred to as replication. While on this side, you notice this extra information obtained from a virus has integrated with the host bacterial chromosome. Now this bacteria B can start showing properties of what the virus brought inside the bacteria. This is the introduction. Now let's look at the concept of prophage that can be induced by a DNA damaging agent. Now how exactly will you decide whether a virus can enter into the lysogenic phase or the lytic phase? This is what we'll be trying to understand as the advanced learning mechanism. Here you'll be having a concept called as molecular switch. As from standard textbook, let's look at this particular zone. The whole area is a chromosome. You're looking at a small area called as genome, which is referred to as right operator. It is referred to as right operator. Onto the left side of the right operator, you have the gene for CI repressor. On the right side of this, we have the gene for CRO. What exactly are they? We are learning about gene regulation. This is gene regulation that we learn in molecular biology of biochemistry. We'll try to understand this, how it makes a difference in the expression of genes in a bacteria like E. coli. Now watch this very carefully. See, what are you looking at? The first time when you see here is the operator in this region being shown in very classical detail. If you focus only on this area, if you zoom that area, you will be having OR1, OR2, OR3. You notice the OR1 is towards the site of gene for CRO. The OR3 is towards the site of CI repressor in this area. Now when I try to use the A gene, the DNA, and try to go for a transcription to produce mRNA, then this is the kind of mRNA that I'll be noticing. So let me erase the annotations here. I hope you'll be able to see it clearly. So what is OR I told you? The OR is right operator, and on the DNA you have the gene for the CI repressor, and on the right you have gene for CRO, CRO repressor, sorry, CRO protein. Now here the OR's area has been properly zoomed and showed into you. You are looking at the mRNA portion here. Now OR1 is towards the site of CRO and OR3 is towards the site of repressor mRNA. Now you will be having the CI repressor promoter coming in this area and CRO promoter coming in this area. Now if you watch very carefully, OR2 will have an overlap from that of the CL repressor promoter. The CL repressor promoter will be over OR3 and OR2, while CRO promoter will be over OR1 and OR2. This is why this particular lines matter. So there is something called as overlap. Again, let me erase the annotations for you to see this better. So I repeat, at this level, you have the CRO promoter and the CI repressor promoter. The CI repressor promoter area will be having OR3 and a portion of OR2 overlapped. CRO promoter will be having OR1 and will be having a portion that is overlapping onto OR2. Now, why am I teaching you all these things? The operator is a region of the viral DNA, some 70 base pairs long. The operators that we are seeing are 70 base pairs long. I said OR right, which is right operator. The whole right operator is supposed to be 70 base pairs long. Okay, when the operator region is enlarged, I told you we have OR1, OR2 and OR3, and approximately what are the MCQs that can be raised? All ORs, each ORs 1 to 3 will be 17 base pairs long, and the remaining areas will be the repressor mRNA and the CRO mRNA. We are seeing whether the repressor mRNA will be activated for transcription or the CRO promoter will be activated for transcription. This is what is our concern. Now, they are the recognition sites to which both the lambda CL repressor and the CRO proteins can bind. So, these are the areas where the lambda repressor of CI and the CRO proteins can bind. Why should we know this? You have something called as recognition sites, right? I'm telling you these areas will be the recognition sites. The recognition sites overlap two divergent portions as I showed you right now. Now, when the RNA polymerase comes and binds here, the recognition sites overlap two divergent promoters, sequences of bases to which the RNA polymerase binds in order to transcribe these genes into mRNA. Now, what happens if I have a controller protein like CI repressor protein being present here? If this is attaching here, then you will be having mRNA for the repressor protein being activated. And if at all there is something binding here, the regulator proteins are binding here, then the CRO mRNA will be activated. So, in order to transcribe these genes, I can transcribe the gene for CL repressor, if at all the promoters and the recognition sites have been activated on this side. If the gene for CRO has been activated because of the promoter region being activated, then CRO mRNA will be synthesized. I hope you have understood till now. Okay. Now, what exactly happens at a bigger level? Now, we are looking at the schematic molecular structure. Here, this one would be the grouping of these kind of proteins. Now, here we have the chain folds here. This is called as the carboxy terminal. This is the amino terminal. Between the carboxy terminal and amino terminal, the linkage is very thick, and this thin linkage can be broken in an easy way. Now here you have to understand the COOH and COOH can come together in this area, while the amino groups are only mildly touching each other. And when this particular protein is capable of coming and binding to OR1, you will be having an OR1 binding domain shown on the amino terminal side. This amino side will be having a domain which can bind with the DNA portion. The OR1 is a part of the gene. So this will be the DNA binding domain, while this one is the intercarboxyl group binding domain. And the CRO protein will be a single dimer. Here you will be looking at them as a tetramer. So what am I showing you here? What you are seeing here is actually the CI repressor protein. And this one is the CRO protein as a single dimer. The CRO protein can bind to the OR3, while this one can bind to the OR1. Just watch as the flow is happening here. Okay. I said OR1 is towards the CRO mRNA. OR3 is towards the CI repressor promoter, a CI repressor area. But whenever the proteins are happening, the CRO protein will act on the OR3, while the CI repressor protein will act on the OR1. This is what is the difference I would want you to remember and recollect. Is that okay? So watching this clearly again, if at all you focus on the gene of the CRO region being transcribed, you will be having CRO proteins being synthesized. This CRO will be a negative regulator for prophage continuation. If the CRO protein comes into action, lytic phase will begin. If the prophage action, that is the CI repressor, is brought into equation, then more multiplication can happen to be inhibited. What does it mean? Then the prophage continues to become a prophage. It remains at the lysogeny phase. What is lysogeny phase? It is the integration of the new genome into the bacterial genome. It remains as that. Active replication does not happen. While the CRO protein things are actually getting activated, lytic phase will be activated. So remember, in a prophage presence, the CRO promoter should be blocked by the repressor proteins in such a way there is no downstream activation and transcription to happen. mRNA or the CRO protein will not be activated. So this is a negative regulation happening. Why? Because CRO protein will inhibit the continuation of prophage. So in simple terms, we have prophage that is supporting lysogeny. We have lytic phase that is supporting your lysis and multiplication of the particular virus. I am saying CI repressor supports this. CRO protein will support this. Now this CI repressor will be having its own promoter area where the CRO proteins will come and block. While the CRO proteins who are supposed to help in the multiplication will have a promoter where the prophage's area's CI repressor protein will come and block. This is called as cross blocking of each other. Now look at this area. For example, if I want to induce the lytic phase, if I want to create a lytic phase by converting a prophage, how am I converting? I am inducing the prophage to go forward. What do? I offer ultraviolet radiation. This ultraviolet radiation will activate a cleavage protease called as REC-A. Now this REC-A will break open all the repressor proteins and they convert all the tetramers and dimers into monomers. Once they become monomers, they won't be able to bind to the OR1 and OR2. Now can you understand this idea here? When I say OR1 and OR2 are here, the next to them will be the CRO genes. If I look at OR2 and OR3 here, on this direction we have the CI repressor protein. Now I told you in the previous slides, the CI repressor proteins are supposed to be tetramers. CRO proteins will be single dimers. If the CI repressor protein are able to bind on the OR1 and OR2, then you will not be able to have activation of the CRO protein. If CRO protein is able to bind to the OR3 and OR2, then you will not be able to have the CI repressor protein being activated. Okay, So let me erase these annotations here for you. As this Rec A protein is trying to break this open, now the blockage of OR2 and OR1 have been removed. Once this happens, then the transcription starting site will be activated and it can move towards the CRO protein's gene being transcribed and translated. What will the CRO protein do? The CRO protein help in the lytic phase, which is it will cause breakdown of the viral particles. Did you understand this part? So ultraviolet radiation can induce the Rec A protease activity, because of which the repressors who are binding to the OR1 and OR2 can be removed. That is, the repressors are broken down, because of which this blue colored island area called as RNA polymerase will be able to move forward in this direction. Now look at this picture here. So in simple terms, we have RNA polymerase here, RNA polymerase here. If the RNA polymerase is moving in this direction, then CRO protein will be activated. If the RNA polymerase is moving in this direction, CI protein will be activated. And this is being controlled. How am I controlling? Because of the gene area. So look at the CRO proteins here and look at the dimers and tetramers here. If it is lysogenic cycle, as you saw in case of the prophage in this particular slide, you will notice the CI protein induces CI gene transcription. How? Because it comes and sits on OR2 and OR1 in such a way the RNA polymerase will not be able to move in this direction. Further, CRO protein synthesis will not happen. And CRO protein synthesis, that is CRO genes transcription and translation to produce more CRO proteins will not happen. So lytic phase is inhibited. So the RNA polymerase is move in this direction and it will be helping in the synthesis of CI repressor. Remember, CRO protein may not have an activation domain, but the CI repressor protein or the CI region has an activation domain that promotes RNA polymerase binding to its own promoter. Okay, it will not be binding to its own promoter. Okay, look at this part, the situation of a lytic infection. The CRO protein occupies OR3. This CRO protein will occupy OR3, because of which this RNA polymerase from initiating transcription from the CI area will not happen. The RNA polymerase will not be able to move in this direction. So if this is occupying this region, RNA polymerase will not be able to move in this direction. CI repressor protein will not happen. So this is where the CRO gene or the CI repressor gene can have control. So let me give you a short summary of whatever we have learned. The whole thing might be confusing because this is additional new information or advanced information. So let me tell you a straight factor here. We have OR1, OR2, OR3 on the operator areas. The OR2 area can be overlapped on the both sides. On this side, we have the CRO genes. On this side, we have the CI repressor genes. Now, if at all the repressor proteins from the CI are coming into action, CI repressor proteins are present, they always help in the lysogeny. Lysogeny means maintenance of the bacteria intact. The integration of the new virus information into the bacterial information is happening. The virus is not actively replicating. While the CRO gene will be helping in the CRO protein, this CRO protein will help in the active lysis or lytic phase of the virus or the cell. Now look at this. If the CI repressor protein comes and binds, it will bind to OR2 and OR1 in such a way the RNA polymerase enzyme will not be able to move in this direction. So it moves in this direction and produces more and more CI repressor protein. And that CI repressor protein can go back and again inhibit this area, and so you will be able to maintain the lysogenic phase. While if I have a CRO protein present, the CRO protein will bind on OR3 and partly on OR2, because of which the RNA polymerase will not be able to move in this direction. It moves in this direction. More and more CRO genes and more CRO gene transcription and CRO protein translation can happen. So this is how the E. coli can control whether it wants to choose, it goes into lysogeny or it goes into lytic phase. So lysogeny, example, how lysogeny is helpful, look at this part. Lysogenic conversion matters a lot in terms of transduction. For example, diphtheria toxin is introduced into a Corynebacterium diphtheriae at the genomic level because of transduction. So a non-toxigenic diphtheria bacteria can become toxigenic diphtheria bacteria because of the lysogenic conversion. Which other toxin? Clostridium botulinum toxin also is because of lysogenic conversion. These are all phage mediated. What else is phage mediated? Remember, the heat labile toxin of ETEC can be phage mediated. So transduction plays an important role here, and if at all that particular bacteria chooses to go for lysis, lysis is a place where one bacteria can become the home for hundreds of new viruses. Either I can choose one virus becoming 100 viruses. The bacteria can decide to act as if it can be the home for one virus becoming 100 viruses, or the bacteria can accept the new information and turn into a different bacteria. If it wants to turn into a different bacteria by lysogenic conversion, it goes into lysogeny phase. If it wants to generate more amount of viruses, it goes into lytic phase. So this is where we integrate the microbiology's bacterial genetics with that of biochemistry, where we discuss about the CRO gene versus CI repressor gene. Hope this helps you. So let's make sure you crack it. Let's crack it. All the best.